All right. Hi, everyone. Thank you for having me today. I'm Carrie Smith, an APHL Bioinformatics Fellow here at the Georgia Public Health Laboratory. And today I'll be talking about the project I've been working on, and it's a validation of a whole genome sequencing assay for respiratory syncytial virus. And so for today's outline, I'll be introducing what is RSV, as well as the purpose of the project, why are we doing this, and I'll walk you through the methods part that includes the wet lab and bioinformatics components, as well as the preliminary results and the next steps in the process. So I will make a note that this is still an ongoing validation and I have yet to complete it. And so for introduction, as we know, RSV is the respiratory syncytial virus. It's a negative um, sense RNA virus, and it is one of the big major three respiratory pathogens that we see every year, and that includes COVID, flu, and RSV. It is acute, it's seasonal, it has common flu-like symptoms. We see cases rise in the fall and winter months. For severe cases, it does cause lower respiratory tract infections, and that includes pneumonia, bronchiolitis. And the vulnerable populations that are impacted are infants, immunocompromised people, and the elderly. Thankfully, in the past few years, we've seen that vaccines have been popping up. So they are FDA approved and readily available for pregnant women and the elderly, as well as nirsevimab, which is a monoclonal antibody that protects infants from RSV. In vaccine research and development, we do see that um, it involves the F and G gene regions. So a lot of the time we see research really focused on like the F gene and the G gene, which codes for fusion and glycoproteins. And the genome size is about 15.2 kilobases, which is about half the size of COVID, and it has 10 genes encoding for 11 different proteins. And the two major subtypes that we do see is RSVA and RSVB. And so why are we developing and validating an assay for RSV? Well, here at GPHL, it is the first time that we are sequencing RSV, and we need to implement a robust and validated whole genome sequencing assay. We also want to develop and validate a bioinformatics pipeline that would analyze the raw reads that come off of the sequencer. We want to be able to detect and subtype RSV confidently. We want to perform near real-time genomic surveillance here in Georgia, as well as track the evolution of the virus and transmission patterns. And this is a very big public health burden, not just in Georgia, but globally. And here's a really pretty graphic of just a general NGS workflow. Um, from last week's talk, Majdi gave a great presentation on explaining the different NGS approaches that one may do, and that would include metagenomics, hybrid capture target enrichment, and PCR amplicon sequencing. And so for GPHL, we went with a PCR-based amplicon sequencing approach since it is very well established, it's cheap, available, and sensitive and specific. So here, for the general workflow, we start with nucleic acid extraction of our samples that go right into library prep. And once we get our library pool, we pop it on the sequencer. And once we get our raw reads, we put it into our pipeline and we get our analysis. Going into the wet lab methods, I'll break down the steps of what I've done so far. First, I developed a validation plan using CDC's NGS Quality Initiative resources. And I will say, anyone doing an NGS validation, I highly recommend checking out these resources, super helpful. During my initial literature reading and going through publications, looking at SOPs, we also reached out to Minnesota Department of Health one of our PGCOE partners, and they were super kind to share their SOP on RSV sequencing. And from there, we finally went and adopted the Illumina COVID-seq protocol with COVID-seq test kit, just because we were already sequencing COVID, and it was very easy to just adapt into RSV. And all we did was switch out the primer sets. As mentioned before, we're doing a short read tiled amplicon approach using the Arctic RSV primers. And here in this image, I just wanted to show an example of the primal scheme output that one may use to design your own primers. Primal scheme is just one of those tools, pretty commonly used. And you can see the tiled image, so overlapping amplicons on top of each other in order to get the whole genome. And these amplicon sizes are about 400 base pairs in length with a 40 nucleotide overlap. Let me see here. The platform we're using is the Illumina NextSeq 1000 and 2000. And for the validation, the comparator method that we're using is the Thermo Fisher RVP assay. It is the respiratory viral panel. It is a pre-amplification PCR assay that detects 41 different respiratory pathogens, and that includes RSV A and B. And this will be our ground truth for validation. And for our acceptability criteria, what would be considered a passing result would be the consensus sequence. Is it at least 90% coverage at 20x depth for base calling? And is it at least 100x average depth across the entire genome? For the validation parameters, we're calculating accuracy, sensitivity, limit of detection, specificity, positive predictive value, negative predictive value, as well as precision. For the bioinformatics methods, we developed a custom Nextflow pipeline, and it's reference-based. And what I mean by that is that we take a reference genome for RSV-A and RSV-B, and we align those raw reads to them and generate a consensus from that. And I'm super proud in this image right here. It took way longer than I expected, but it does give a nice layout of how the pipeline works. It's divided into 3 stages. First stage is pre-processing, so that includes our quality control, our adapter trimming, human reads removal. Stage 2 includes k-mer detection, reference selection using BBMap, and how BBMap selects the correct reference between A and B is that it'll map the raw reads to each reference, and whichever has more mapped reads, more positive hits, it's going to select that reference. And then that'll pipe into the BWA alignment step, generating the BAM file, which then gets processed through SAMTools and IVAR. And finally, stage 3 includes consensus generation and variant calling using IVAR, coverage and depth using most depth, lineage classification using Nextclade. And that is where, with Nextclade, I combine all the consensus FASTA files and pop it into the Nextclade web browser, and it generates those lineages as well as a phylogenetic tree. And I use multi-QC to give a final subreport of all the data. And then all of this is done in our AWS Health Omics environment using Docker containers for each tool. And for code review, for version control, all the scripts are placed into a private repository in GitHub. For the preliminary results, the clinical specimens are nasopharyngeal swabs we use, and they're positive by RVP if they had a CQ of less than 31. I selected 44 RSV-A and 44 RSV-B for the sample set. Um, for the controls, we use a synthetic transcript control from Zeptometrics, one for A, one for B. And for the negative template controls, we use molecular water. And you see on the right in this table, PCR represents the RVP assay, and that's our comparator, our ground truth. And WGS is our test method. And we can see that most of these samples are in agreement between each other. And let's see. However, oh, sorry, to back that up, I noticed like coverage started to drop off after a CQ value of 20. So we established a cutoff at 20. So the table represents a subset of all the samples I've sequenced. So the subset contains 28 RSVA and 24 RSVB samples. I do want to point out for those samples resulting negative for WGS, so in these cells right here, I wonder if I could laser pointer here. Here in these cells, this could be due to viral loads being too low for sequencing. So that being said, because I haven't established an LOD quite yet, so I want to emphasize too that the CQ values I'm getting from RVP is not true CQ as they are lower than expected, just because RVP goes through a pre-amplification step. And that means, for example, if a sample has a CQ of 20 from RVP, it could be a true CQ of 30, just to put that out there. So in this case, validation parameters that we see right here, they may be affected. And that poses unique challenges to this validation. And that includes, we're not able to determine viral concentration directly from RVP-CQ, and therefore we're not able to establish an LOD from RVP clinical samples. But nevertheless, after further discussion, we're able to come up with a solution. So for the next steps, I'll be ordering a couple culture specimens of known concentration from a vendor for RSV A and B. And with those known concentrations, I'm able to serial dilute them and then determine the LOD in copies per mil. I also plan to resequence some of the samples that need confirmation. And once I'm all done with that, I'll run the precision experiments that will cover repeatability and reproducibility. And also, if once all validation metrics pass and there's no need to repeat anymore, I'll write up a final report. And that concludes my talk. I like to give a huge shout out to everyone involved in the project at Georgia Public Health Laboratory. I like to give a big thank you to my mentor, Dr. Amaya, as well as Dr. Parat. I'd like to thank Dr. Kirsch, Dr. Heck, as well as Blake and the virology team. A special thank you to Dr. Nandu Balakrishnan and Jason, and thank them for their expertise and guidance in the project. And of course, last but not least, I'd like to thank the bioinformatics team, Tatiana, Harrison, James, helping me through any troubleshooting I had and getting my pipeline to run successfully. And with that, I'd like to thank you. And I guess, I don't know if we'll do questions later, but yeah.
Yep. Thanks, Carrie. So what I'm going to say is if you can stop sharing and then
Sure.
Yi Huang can go ahead and start sharing hers, and we can maybe take one or two questions just real quick if anyone wants to chime in while we're doing the transition in slides here. Or if you're feeling bashful, you can type it into chat. All right, I'm not seeing anyone go real quick, so thanks again, Carrie. And let's go ahead on to presentation number 2, Yi Huang from Florida.
Thank you for the introduction. Can you see my screen?
Perfect. Yes.
Okay. Okay. Hi, so today I'm using two sequencing cases to present the short read and long read sequencing application in our molecular lab. We have learned the principles, method, and the differences for long read and short read sequencing in previous seminars. In this series seminars. So I will use the applied cases to demonstrate more details for these two sequencing technologies in following sections. I will summarize the procedures for the sequencing work. The short read sequencing will be presented with TB. For the whole genome sequencing application, the long read sequencing will be discussed with Candida auris, whole genome sequencing application. And then there will be comparison and summary. So since I don't have a camera, let me briefly introduce myself. I have a PhD in plant pathology from the University of Florida. I started working at the Florida Department of Health as a medical laboratory scientist, focusing on sequencing laboratory work. Now I work as a bioinformatic data analyst supporting whole genome sequencing data analysis. This gives me experience working in both wet lab and dry lab environments. Today, I will discuss TB and Candida auris WGS based on my own hands-on experience. So, quick summary. In our department, we use WGS to monitor infectious disease outbreaks, to identify pathogens, to predict antimicrobial resistance, and for routine pathogen surveillance. For both long read and short read sequencing, they all include following procedures: DNA extraction, library prep, and sequencing run, the bioinformatic analysis, which we performed on Hypergator. And normally there will also be epidemiological analysis following the outbreaks. So let's start with TB. TB has been a challenge for public health worldwide, and the drug resistance has been rising and there have been so many deaths from TB in 2023 based on WHO. So the monitoring for outbreaks and the drug resistance detection are important for public health surveillance. So we already know that WGS can help monitor TB transmission and identify resistance genes. Short read sequencing has sufficient capacity for our current weekly workload of 20 samples due to the genome size of TB, which providing adequate depth of analysis. And the variants related to drug resistance have been well studied with clear documentation, which make the resistance detection relatively straightforward. So before the sequencing work, the TB clinical samples are collected from the ordering facility. Then the TB department will coordinate with APIs on the needed test. They will run identification and drug susceptibility test, and culture and stock the samples. Then the necessary samples will be sent to our part for WGS. So when we receive those samples, which will be pure culture as shown in the picture, we start with bead beating by using this instrument, Quick Prep, and extra DNA by using XAML Research Kit. That required the required size for MySeq, which we use for short-read sequencing, is around 300 to 500 BP. And normally the concentration of DNA after extraction are more than enough for the sequencing. So in this procedure, it's not critical for controlling the size and the concentration. Here to mention, there's also alternative sequencing method by using diplex to detect 18 known resistant genes, which using clinic samples directly instead of pure culture by targeted sequencing. The MySeq workflow, which is the next step, begins with library preparation, where DNA is fragmented and repaired and ligated with index adapter for pooling. After loading in the instrument, onboard cluster generation creates thousands of copies of each DNA fragment through bridge amplification, which we have learned from previous seminars. Sequencing by synthesis incorporates fluorescently labeled nucleotides one at a time and capture the signals to base calling during the run, which generate FASTQ files with quality scores and the multiplex index samples. After sequencing is complete, we then download the FASTQ files from each project from BiSpace through Hypergator. Then we use a pipeline developed by CDC called WordPipe, which includes assembling and variant calling steps to check 18 loci for drug resistance detection. This pipeline has QC at the initial step, with threshold of 30 for the depth and 95% for coverage. If a sample fails QC, it will not proceed to the following steps. And we also use H37RV as a reference genome for mapping, and we include it in each round as a positive control. Here's an example for the output files after the pipeline. Here's lineage report, and we have annotation report, and also there will be a summary report for each sample. Here is the example for show you what the output looks like. So we can gather information including the depth, the reference genome coverage, and the detected mutation loci. In this case, after detecting one gene locus, it determined that this sample is resistant to isoniazid. Next, I will talk about Candida auris. As a multi-drug resistant Candida species, it has emerged as a causative agent of many healthcare associated infections and associated with outbreaks. The cases have increased since It first found in US in 2016, and it can rapidly develop antifungal resistance. Meanwhile, it's easily to spread and hard to kill by commonly used disinfectant, if you remember the movie The Last of Us. Why we use long-read sequencing for WGS? We already know that WGS is more accurate to identify the pathogen and can track the clades and transmission patterns for outbreak investigation. Also, with a genome size of 12.4 MB, Candida auris is well suited for long-read sequencing using PacBio technology, which has high throughput. Multiplexing samples provides sufficient sequencing depth for comprehensive SNP detection and improved genome coverage. This approach enables efficient weekly surveillance run for outbreak monitoring, which is 20 samples per week. Since we need to study and predict the variants, PacBio has the ability to produce less systemic errors and more complete assembly. It's better for identifying gene location and complex variants. So same as TB before, similar. Before sequencing, Candida auris samples are sent to Tennessee, and they pick 20 to 30 samples that should be sequenced depending on outbreak priority, then sent back to us. So our HAI team will run identification, drug susceptibility test, and culture and stock those samples. Then the certain samples will send for WGS. So the workflow for long-read sequencing lab prep, including size control, barcoding, and pooling by using smart build kits from PacBio. For size control, which is a critical step, start at the DNA extraction step. The isolate is collected from pure culture. Instead of bead beating, we digest the sample using Zymolis because PacBio requires DNA fragment size of around 10 kb for optimal output. And bead beating would not maintain those fragment lengths. To control fragment size, we use mini-G, which is this instrument, to share the DNA in the setting of 1500 RPM for 2.5 minutes and achieve the ideal length. Since there's no amplification step during sequencing, the required input DNA for PacBio sequencing is around 1 microgram. This can be challenging because Candida auris DNA extraction efficiency is already low. We typically obtain around 1 microgram DNA per sample, but after laboratory prep and multiple purification steps, the final yield may be like way less. It's 1/3 of the original concentration. But we have heard that PacBio has newer products requiring lower DNA input for sequencing. So hopefully this critical requirement will be improved in the future. But so far, this is a delicate step, and we try to maintain the highest input for the running. After size control, following by DNA end repair and A-tiling, and ligating smart bell adapters also index to create circular templates. So after that, the prepared library are then loaded into the smart cell, which containing 1 million zero-mode waveguides where DNA polymerase incorporates fluorescently labeled nucleotides in real time to generate continuous long reads. And the circular consensus sequencing algorithm process multiple passes to create the hi-fi reads after 30 hours run in our setting. After the run finished, the BAM files, this is example for one sample. As shown here, they are transferred from the server connected to the PacBio instrument for storage through Hypergator. The pipeline Pensacola, which was developed by Ibo from our team using Nextflow, is used to process the data analysis. So this pipeline including QC, de novo assembly, reference assembly, identification and species abundance detection for contamination screening, and variant calling for drug resistant detection. And for now, we're using the original genome B8441, but I know CDC has the new assembly with better quality. So we plan to change the reference gene as well. And here is an output example showing coverage and depth calculated for 7 chromosomes of Candida auris with taxonomy identification. We also have a list of drug resistance related gene loci to check for mutations and corresponding drug resistance patterns. We constantly achieve satisfactory coverage and depth from this sequencing. And PacBio long read give us fewer contigs compared to short read, indicating good assembling quality. Here is a study I conducted last year where 100 samples were sequenced for phylogenetic and drug-resistant analysis. So interestingly, we found samples belong to only 3 clade. Clade 1, clade 3, and just 2 clade, sorry. With drug resistance to 5-fluorocytosine and azoles detected. The samples was from the clinical samples and the CDC samples for the clades in the identification. This demonstrated that using long-read sequencing, we can conduct comprehensive studies focusing on phylogenetics, isolate evolution, and drug resistance prevalence. So to summarize the difference between long-read and short-read sequencing, for input genomic DNA, long-read sequencing requires more critical size and quality control, where short-read requirements are easier to meet. Long-read sequencing doesn't require PCR amplification, so its errors are non-systematic and random, and can correct it with increasing coverage. Therefore, assembly are more complete and structurally accurate, which better to identify the gene location and resolve complex structural variants. And in conclusion, both technologies have their places in public health lab. The choice depends on the specific goals, the budgeting, and the complexity of target organisms. So that's all my presentation for today, and I'm happy to take questions if there's any.
All right, great job. So I'm going to do suggest the same thing as last time. So Yuong, if you want to go ahead and stop sharing your slides while we answer any questions, and there's one on chat for you. And then Madhav, if you want to go ahead and start sharing your slides while she does that.
Okay, thank you.
All right, and so maybe what we'll do is we'll let the Q&A go on in the chat on the side while we go ahead and move on to Madhav here, who's going to be our last presenter, talking about salmonella, and Madhav is from the Mississippi Department of Public Health Lab. Sorry.
Hi, can you all see my slides?
We can.
Great.
But we're seeing the presenter mode. So
Oh,
if you switch
let's see.
screens, that might be great.
How about now?
Perfect. Yep, great.
Okay, great. Hi, my name is Madhav. I'm an epidemiologist at the Mississippi Public Health Lab, and I'm going to be talking to you guys about a molecular epi case study. So we sequence salmonella as part of the PulseNet project. So I'd like to start with just a brief introduction to PulseNet. PulseNet is a national laboratory network which standardizes sequencing data for foodborne outbreak tracking. And the way they do that is the CDC sets guidelines and workflows for sequencing, analysis, and cluster detection. This data is uploaded real time to PulseNet's national databases with demographic data per sample. And then PulseNet uses comparisons of that data to monitor for national outbreaks and notify states regarding those national outbreaks if their samples are related. PulseNet is made up of laboratories in all 50 states and various regional territories, such as Puerto Rico. They also partner with some food regulatory labs, so the FDA and the USDA. We also have some international partners through PulseNet, and there's the global PulseNet International Lab Network. PulseNet consists of diseases like Salmonella, E. coli, Listeria, Vibrio, and a few others. And so the background is we sequence for this project, and that's how we are involved in tracking foodborne disease. This investigation began in November of 2024 when our EPI team was informed of several individuals who became sick after eating at a restaurant, which we'll call Restaurant A. They were informed of this because of complaints made to our Office of Food Protection. We then reached out to the EPI team. The EPI team very quickly identified 6 related samples which had been submitted to the public health lab from a hospital A. And they contacted our sequencing lab and asked that these samples receive higher priority for sequencing. And then within a week of that, they identified 2 more samples, making it 8 in total that they believed were all related. And these samples were also sent to the public health lab, and all of the testing was prioritized. Okay. We have 2 broad sequencing options at the lab. We have an Illumina instruments, which are made up of the MySeq, which is our primary sequencing instrument. It can sequence about 24 samples per run. We also have an iSeq 100, which is used for faster turnaround times, but has a much lower output. We can only do about 5 samples per run. We also have a NextSeq, which is a high sample load. It can do about 100 samples per run, but it's difficult to library prep for, and so we don't use it unless we have such a high sample load. Our primary sequencer is the MySeq, and we have 3 of those that we use. We also have a ClearLabs instrument, the ClearDX. This instrument fully automates the NGS workflow from extraction to the end product. And we use it a lot for cluster or outbreak related samples because it's so easy to load the samples. And it can do about 10 samples per run. Importantly, it uses 2 iSeqs to do the actual sequencing. So we have 2 different workflows. Actually, we have a manual wet lab workflow. This is what we use for the MySeqs or the other Illumina sequencers. Sorry, guys. So for this, we accession samples and then replay for growth overnight. The bacterial DNA is extracted manually using a DNase blood and tissue kit, which is made by Qiagen. We prepare the libraries using the DNA prep library prep kit, and then we pull them and load them onto one of the 3 sequencers. The ClearDX workflow is much easier. We accession and replay it overnight, just like for the manual. But then all we have to do is add bacteria to tubes with lysis buffer and beads, vortex for 30 seconds, leave it to incubate for 30 seconds. Take the liquid from the tubes, add it to a sample plate, and then load the sample plate and any other reagents onto the instrument, and it does the rest. Our dry lab workflow begins when the sequencing completes, which usually takes between 27 and 40 hours from loading the sequencers. We have FASTQ files which are generated, and those FASTQ files are submitted to PulseNet 2.0, which is this online GUI-based analysis platform that I'm sure many of you are familiar with. This is used for analyzing the sequence data, and we also feed it metadata related to the samples, which is used for cluster tracking. PulseNet 2.0's pipelines perform speciation, QC, and serotyping. And then all samples which pass QC, we submit to the national database that PulseNet monitors and keeps, as well as to NCBI. PulseNet assigns something called an allele code to all of their samples that are submitted to the national database, and that's what we largely use for cluster detection. So what is an allele code? An allele code is is a code which gives you an approximate difference threshold. So you have a code at the very beginning which tells you the genus of the sample, and then each digit represents an approximate allele threshold. So for Salmonella, for example, the 4th digit represents 7 allele differences, and so if 2 samples share the allele code to the 4th digit, you know that they're probably related somewhere in that ballpark, within 7 alleles by core genome and LST. PulseNet defines a Salmonella cluster as 3 or more cases collected within 60 days of each other, which are related within 10 alleles by core genome MLST. And 2 of those samples have to be related within 5 alleles. PulseNet, an important distinction between a cluster and an outbreak is that a cluster must have a common epidemiological source or cause, which is identified. Otherwise, it can only be called a cluster. A cluster is samples that are genetically linked only, and they can't be called an outbreak until you have a common cause. So after we sequenced those 8 samples, we had all 8 of them identified as Salmonella serotype enteritis. PulseNet 2.0 is really convenient in that it generates a distance matrix and a dendrogram. And when we looked at that data, we found that all of the samples were closely related within 0 alleles. As a follow-up, we decided to also compare these 8 samples to all other Salmonella enteritis samples collected in 2024. And follow-up analysis identified that 3 samples from the second facility, or from a second facility, Hospital B, which is located in the same county and city as Facility A, were related within 0 alleles also. All 11 of these samples we've noticed were from people who lived in the same county, which we'll call County A. And this is what the initial dendrogram looked like when we took a look. And you can see that everything is very, very closely related to this single, vertical line. It gave us a reason to say, okay, there's definitely something going on here. As the next steps, we immediately notified our epi team that the samples they requested were related genetically. We also notified them about samples from Facility B, which are notified. And so we decided to give priority to all samples that we were receiving from hospitals A and B, and also give priority to any patient who lived in County A as we continued sequencing. And in this process, we identified 24 additional cases which were related. And in total, we ended up identifying 35 samples in total that were closely related. This is what the final dendrogram looked like. It's a very tight cluster. All of the samples were related within 0 to 2 alleles. I think only 4 of these samples were from people who lived outside of County A. The rest were all people who lived in County A. And importantly, this is still a cluster. We can't classify it as an outbreak at this point in the investigation because we haven't identified a direct epilink, or at this point in the presentation. So while we were doing the sequencing, Epi had continued their investigation. Our epidemiologists performed an initial foodborne illness investigation to identify any potential exposures and common symptoms. An outbreak-specific questionnaire was also then developed, which they used to interview diners at Restaurant A, since that's where the initial complaint came from. And in doing so, they found that all 35 of the people who were sick had eaten at Restaurant A. And then they developed descriptive case statistics and calculated food-specific attack rates from the questionnaire data, and also sent an environmentalist to Restaurant A to perform an on-site hazard assessment to identify any potential issues that could be there. The epidemiologists found that 2 food items were found to be significantly associated with the illness. There was a meringue and banana pudding. The meringue had a pretty high food-specific attack rate, about 91.4% in exposed patients versus 13% in unexposed patients, which is an attack rate difference of 78.4%. They also found that the relative risk of getting sick from meringue was about 7.03 with a p-value of 0, which is very statistically significant. The banana pudding had a much lower relative risk, 2.23 with a p-value of 0.0001. Which led our epidemiologists to think that, the meringue might be the main issue. And 32 out of the 35 identified cases had reported eating meringue. The environmental assessment also noted that there were eggs in Restaurant A's refrigerator, which were dirty, covered in feathers and debris, which further solidified evidence that the meringue was probably the issue. And at this point, now, because we have epi data and genomic data, we can say this is an outbreak. The epi data identifies that meringue is the likely causative agent for this outbreak. As a result of this investigation, our epi team reached out to the restaurant. Restaurant A placed a voluntary hold on eggs. They substituted the use of pasteurized eggs for future meringue recipes, and kind of eliminated purchasing eggs from the farm that they'd been using. And those changes resulted in no further associated cases since they implemented the changes. I'd like to make a few acknowledgments. I'd like to thank Beata Krylowicz and Sehu Sise, who are my colleagues in the core sequencing lab, for all of their hard work. We'd also like to thank Carla Boutwell, Theresa Kittle, and our Office of Epidemiology for all of their collaboration, as well as our special microbiology lab for their help in getting salmonella positives. This is what the dirty eggs would have looked like. This is just a picture we found, but as you can see, there's definitely a high risk of illness with something like that being used in a restaurant. And do you have any questions? I can take those now.